Hi, Bill Mobley at the ANA meeting speaking with Soma Sengupta from the Farber Center and from the Mass General Hospital, Dana-Farber Center. And so, Soma, welcome, and we want to hear about your very interesting research.
Hello. So basically I was fortunate to receive an R25 NINDS during my residency at Beth Israel Deaconess, and the award was awarded to both Beth Israel and Children's Hospital Boston. And I worked on a project which involved membrane proteins, which is something very dear to my heart. My scientific training is with membrane proteins. Mm-hmm. And using the sort of genomic data that was obtained from Dr. Pomeroy and other colleagues, we saw that in the most aggressive subtype of medulloblastomas that express photoreceptors, MEC, we noticed that there was a GABA5 signature. And Dr. Yoon Jae Cho, who was in Dr. Pomeroy's laboratory, was almost like a mentor to me in this project. And we started working on this GABA5 idea, and I took my sort of membrane protein background and had a lot of fun doing this project.
Good. And so what did you find out?
So we found out that GABA5 can be used to modulate the tumor. So we found a compound that inhibits the cells that contain GABA5 selectively over those that do not. And we were able to reproduce these effects in an animal model, but we're still studying the in vivo effects.
Very interesting. Is it an antagonist of this receptor subtype? Or?
No, ironically, it's actually an agonist.
Uh-huh.
So what we think is happening is that the neuromodulatory properties of the GABA receptor and the drug keeps the channel open for longer than anticipated, and then the drug binds, letting signaling molecules things in, and then somehow the cells get destroyed in vitro. In vivo, however, the effects are not prolonged, so you do need a second agent, like a chemotherapy agent.
Very interesting. So are medulloblastoma cells immature then with respect to chloride gradients? I mean, are you exciting the medulloblastoma cells and killing them? What's your thinking about that?
Okay, so we did quite a bit of electrophysiology, both on single cells and also in ex vivo tissue slices, and we found that that basically there's a very high resting membrane potential, so it doesn't act quite the way as you'd expect. And so Frances Jensen, who was the other mentor in this project, and her postdoc Hong Yu Sun, did the beautiful electrophysiology work for this project.
So the agonist, then how does it affect membrane potential?
So basically it makes it, so at resting potential the membrane is very negative, and somehow it makes it less negative.
Interesting.
And then the drug binds, and then you basically have it open for much longer than normal.
Very nice. Beautiful work.
Thank you.
We hope it makes a real difference in the lives of these little kids with medullos, so congratulations.
Thank you very much.
Thank you.